Now let's try this. Last time we tried this, can we turn the lights off, please? Last time we tried this, things got off track a little bit. Hopefully we can get back on track. I love your hair. That's nice. All right, so there was a question as to what we did on Friday. I thought it was clear what we were doing, but I guess a lot of students just didn't get the message. I don't want to discuss it anymore. I tried to discuss with you this idea on Friday, if you remember correctly. Try to discuss what you're doing, even though you aren't intrinsically interested. What do I mean by that? I mean, there's a lot in life as you become an adult that you have to do even though you're not interested in the topic. I can make a long list. I'm sure you and your folks could go home and discuss it on all the things that they do on a daily basis they're really not interested in doing. But they have to do it. Paying the bills, et cetera, et cetera. They're just things that sometimes, it's not, maybe you're not a numbers person, maybe you're not, maybe you just don't like reading, but you have to read, I don't know. Whatever the case is, there are things that adults have to do that they're not intrinsically motivated to do. And I think we went over, please correct me if I'm wrong, we went over some stories on Friday. Is that right? So in any case, we discussed various topics on Friday, trying to focus on the idea that we have to do even when we're not interested in it, and what is appropriate behavior under different circumstances. Is that correct? That's generally what we talked about. All right. We talked about this idea of growth conferences and how we have to try to get through these growth conferences. Is that correct? Yes. And I gave you 3 documents. Is that correct? Yep. That's right. One of those documents was an outline of how to do Punnett squares. Is that correct? Yes. And it gave you all the definitions that we've been covering. I need everyone on the same page. So I don't want anyone having side conversations. I need you all focused and either answering, nodding, somehow representing that you do agree. If you disagree, please raise your hand and say that's not true, that's not what happened, and let's have this discussion. Please and thank you. So on Friday, you got 3 documents, and they were on Punnett squares, stuff we've been covering now for 2 weeks. We went through mitosis and meiosis, did we not? Yep. And we went through meiosis and we said what happens in meiosis in detail, correct? Take out your Dragon Genetics packets, please. Something that you've been working on now for 2 weeks, I believe. A week? All right, only a week. Okay. Only a week. I've done it for a week. You've uploaded it, correct? So you've answered all the questions.
No, I need help on the back page.
You need help on the back page. Very good. We're going to focus on page 2. That everybody here, is everyone done page 2? Page 3, is everyone done page 3? Yes. And page 4. That's what we're going to focus on today, because that's what your Punnett squares are about and how we do it. Thank you. You're welcome. In class, I gave you we went through an entire lesson on how to do Punnett squares, did we not? I posted it online. We looked at all the different options, all the genotypic ratios, all the phenotypic ratios. We said you get 1 to 2 to 1 to 2 to 1 to 1 to 2. Remember that? And 9 to 3 to 3 to 1 was the phenotypic ratios. Do you remember that or not?
Yeah, no, I do not remember.
Like, I remember briefly, but not really.
So you didn't review that in your notes. Do you have it in your notes? You have it in your notes? Good. Do you have it in your notes?
Yep, right here.
All right, right there. So so now you wrote it down. That's the writing part, that's the recording part of our Cornell notes, but you're not reviewing. You're not revising. Because if you were revising, you would have taken that information, and as you did the what is it you said, a week of Dragon Genetics? You would have taken that Dragon Genetics and the information you learned during that activity, and you would have added to your notes. Does that make sense? And if you didn't understand something, you would ask questions. Ask questions. You would, during the activity, before the activity was due, right? You would revise your notes as you did the activity. You would go and review your notes. If you missed something, you would watch the video and you would bring in specific questions concerning the notes, concerning the video. When might you ask such things? Well, one would expect that you would ask things before class, before school, during lunch, and after school. Or you would go to tutors. Or you would go to Google. Or you would go to your chapter reading. So those are the expectations that we have in high school. That when you would either go to see the teacher during their office hours, and I believe the professor on Friday discussed with you kind of expectations of what you're expected to do in college, correct? Do you remember that? Yes or no? Yes. The professor from Notre Dame came here, spoke to you for an hour, discussed with you what you're expected to do when you get to college, how can you be successful when you get to college, how you take notes, how you revise, how you see a professor or tutor during office hours. This is twice a week. The tutors are here from Case. Google is actually 24/7. And chapter reading is 24/7. You have access to that. I emailed you the chapter. I sent you the chapter in a PDF format. So you can have it on your phone anytime you want. You can download and put it in whatever app you have. If you're an Apple user, you can put it in your iBooks, whatever you need to do. So those are kinds of the things that I would have expected from you as a student when you're not understanding something, when you have a question on something. If you didn't have any questions, you didn't have to do all these things. You can choose. But you have to do something. You can't expect to do well in class without doing something. I could sit here and explain how to do these practice problems I've given you. In fact, I have here from the previous class, I did go ahead and explain it. I feel like me explaining it is a waste of time. I feel like that because I've already explained it. I've walked through it once in class. I've given you 3 days of practice in class, in groups, given you questions to answer, walked around and helped you answer those questions. And then I've given you some practice problems with an outline of step by step what to do. If you look at the paper that was handed to you on Friday, if you look at the outline, it has all the definitions and a how-to on Punnett squares. So I guess you could add that. You could reference the handout, which is an outline of, and we'll go ahead and put or. And again, that's 24/7 since I printed it and handed it to you. Is that a fair expectation? Could someone tell me that whether they think that's fair, what I just stated? Yeah.
I mean, it's fair, but in the same sense, like, if we're in the classroom, if you have a question, I feel like, why are we...
Well, you don't have to come after school. I encourage you to ask a question in classroom. As long as the question is what? I want you to be what? What are the things I've said to you about questions? Relevant and specific. Relevant questions in class. I beg repeatedly you to ask them. I tell you, if you're embarrassed, you don't even have to ask them in class. You could send them to me the night before. Like you have questions on the dragon genetics, you could have emailed me those questions.
I stayed up and...
Okay. My point is not you specifically. My point is that's the expectation. So questions in class is what I encourage. It saves us all time. Because if you have a question, I can almost guarantee at least 3 others have the same question. So questions in class concerning the topics, but they have to be relevant and they have to be specific. The question I get continuously, all the time from students, all the time is maybe an exaggeration, but I get a lot. Is this. Are you ready? I don't understand anything. Could you explain it all again? That's not useful. Are we clear? It's not useful. If you have something specific, if you actually tried to do the problem and you were walking through, you're like, whoa, I don't get it. There's something missing here. Write down what that is. That's part of Cornell Notes, isn't it? Correct? Isn't that what the margins are for in Cornell Notes? For you to write down the questions that you have about any particular sample problem. That's what you're supposed to do. Not just write down the question, but then find the answers. Where would you find the answers? Any one of these would do. Ask questions in class. Reference the handout. Chapter reading. Google. Tutors. After school, during class, during lunch, before school, any one of these. Now, the other thing that kids continuously say to me is, you're busy during lunch. Yeah, there's one of me and 120 kids. I guarantee you, somebody needs something, a letter of recommendation, a reference, tutoring. So get in line. So hustle. Find me and use me appropriately. All right. So let's go ahead and move on and take a look at what the content issues are. Do I have any questions, any concerns at this point, right now? So right now what you should have is your dragon genetics out, that you've completed, you've uploaded. You should have uploaded them. If you didn't upload them, you should have showed it to me. You know how this rolls. You want to guarantee your points, you upload them. You need to show me, you come during lunch, before school, after school, and you show it to me, I put it in, you hand it back to you. You know this. There's a procedure for everything. All right. Now, that being said, your dragon genetics should be in front of you. If it isn't, you better share with someone, and you take out your handouts. All 3 handouts should be in front of you as well. So, like I said, saying to me you don't understand anything is not an acceptable question. I need to know specifically what it is you don't understand if you expect me to answer a question. Yes?
You know how to do Punnett squares with the FOIL method? You know how you have 3 and you put them in Punnett squares? I don't understand that. I do it.
Say it again.
So you know you have 3 different genes you have to figure out? I don't understand how to do that in Punnett square.
3 genes. Or 2 genes. Which is it?
So this I don't understand. You have some people who do
How many genes are involved there? You tell me. There are 4 genes involved there? Go sit down. So this goes back to whether you actually paid attention to is correct. Listen to her.
Let me show you. Wait, look. Is the 2 T's or just 1? And then the A.
Yeah?
I get confused finding the percentage. Oh, yeah, for this one. With the, hybrid cross. I get confused finding the percentage.
I'd like to deal with one question at a time if that's okay with you.
Okay.
How many genes are on that example you just asked me about? 2. How do you know there's 2 genes? Look at, a minute ago you were confused about how many genes you had. You told me 4. Is that correct?
Yes,
That was you what?
I was saying that they were correct.
Okay. But you understand.
Yes, I understand.
Don't just say you understand if you don't understand, because you're going to come to a problem again, and you're going to have to know how many genes you're dealing with. So how do you know that there's 2 genes? You tell me how you know.
So normally there's like a dominant and a recessive gene. So you just look for it. Because the dominant is in the front and the recessive is in the back.
It's interesting. I'm not sure what you mean by that. Yeah.
So a gene is with the 2, is with the 2, like, for example, T's. So that would be a gene instead of TA, letter gene.
So that's good. That's the way you need to think of it. Each gene has its own variable. Correct? So you have to think about what exactly is a gene. It's a part of a DNA molecule. Starts at beginning, end, beginning and end. And there's different versions. You get, if one DNA molecule came from dad, the other DNA molecule came from mom. So 2 different versions, beginning, end. Each one could lead to 2 different outcomes. So one could be a big A, the other one a little a. But A is the same gene. It leads to the same, different versions of the same gene product. What is that gene product? It is a protein. A protein. Let's do that. I hear that. So, I was. Right. Okay. So Punnett squares. Actually, is it? Could you? I read. Just bad timing. Please.
She said it right there. I'm sorry.
See, can we not do it? Because I'm sorry, this particular class has an issue, and that particular young lady was was part of that issue a minute ago, and it just caused. Look, see, this is what I got to deal with, and so I know it's all right. This is okay. It's okay.
Did you have a good weekend?
Okay. Are you done? I'm not talking about anything except what I'm supposed to be talking about. So I'd appreciate if you wouldn't interrupt. And if you're going to interrupt, young men, separate now, please. Could you 2 gentlemen separate right now, please? Thank you. Why don't we go ahead and do what we're supposed to do? And let me not worry about what you remember, what you don't remember, and let's just move on. Say what?
You're supposed to be doing great.
I don't care. Away from him. Someplace, not near the chemicals, where I told you you're not allowed to sit. Oh, that's right. Maybe you didn't remember that either. Sit somewhere where you're supposed to sit. Oh, you? Okay. I'm okay. Let's play.
We're supposed to be doing
Wow. Wow. So in dealing with a protein, the gene product is either big A or little a. You're either dealing with getting, I don't know, black or white. You're either dealing with tall or short. Is that understood?
Yeah.
That's what you're dealing with when you're dealing with big A or little a. Those are different versions. So if you have an A and little a and big T and little t, how many different genes do you have? 2. How many different alleles do you have? 4. So you have to know the difference between gene and allele. But that has nothing to do with Punnett squares. That has to do with your understanding of the previous units. Right? You see how this all builds on itself? If you didn't learn what a gene and what an allele was, you don't know what it is now, so you're confused about what's going on. So, moving forward, a very specific question, a very good question, was how do you do FOIL? That's a specific question. So when you're taking a look at FOIL, you have parent T, big T, big A, little a. And I'm going to go ahead and change it and say mates with another parent who's big T, little a, big A. So now you have to understand how does mating when sexual organisms have sex, how do they produce, what do they combine? Do they combine these two cells? Is this going to be combined with this? So this is 2 versions of each gene. 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 Yes? So is this a diploid cell or a haploid cell? Why do you call it a haploid cell? I'm glad you're answering. This is what I need you to do, is to answer specific questions. Why do you call it a haploid cell? Why do you call it a haploid cell? What is a haploid cell? See, this is what, again, the previous unit, half the DNA. Not half the cell, half the DNA. How many versions do you normally have in each of your cells? How many different versions? 2 copies, right? 1 from its dad, 1 from its mom. 1 from its dad, 1 from its mom. Correct? Is this a haploid or a diploid cell? Diploid. Well, how do you know? It has 2 of each. Is that clear? Okay. So it said 2 diploid cells. Some of you are not paying attention to what's going on the board. You're not taking notes. This is how we started this whole discussion. Why don't we all turn around and make sure you're facing the board when we're writing notes? So we have 2 diploid cells. You're making 2 haploid cells. Don't you have to make 2 haploid cells before you can combine them? Didn't we do mitosis and meiosis? Isn't that something we covered in this class? We had a whole unit on it, correct? So you take an egg and a sperm and you put it together. You can't take 2 skin cells and put them together. We talked about that, did we not? So how do you put these 2 together? What do you need to do? Yes, you're right. My question is, what do you need to do in order to combine these 2 cells? Yes. That's right. You got it. You got the mechanism. You're absolutely right. You do FOIL. Start from the outside, move inside. Correct. That's perfect. The question is, what is your objective? What are you trying to do?
Make a big.
We have the alleles. These are the alleles this parent has. These are the alleles this parent has. It's only going to be able to combine what? Can it combine these 2 cells together? Yes? Or no? Pick an answer. Why not? What's confusing about my question? What am I being ambiguous? What's the issue?
What does that mean? What that mean?
I'm not specific enough in my question?
No, it is.
Like I'm asking you, this is the one parent. It's a diploid cell. This another parent's a diploid cell. Mm-hmm. What do they have to do if they're going to make a baby using sex? You're going to combine these 2? So let's combine them. What?
half.
Say it again.
You have to reduce the DNA to half because if you made it just like that, it's going to be double the chromosome.
Right. You have to reduce the DNA by half. You have to do meiosis. You have to reduce it by half. That's the whole point. That's why we went through meiosis. You have to make an egg and a sperm. You have to reduce it by... Am I clear? I don't want to have to say this. I've said this for weeks now, right? We've done meiosis and mitosis. We have to reduce the DNA by half in order to combine them into a new person. Because if you tried to combine them the way they are, you'd get 4 T's and 4 A's, and that's just too many. Is that correct? So how do you reduce this by half? If you have 2 T's and 2 A's, what do you have to have?
1 T and A.
You have to have. Now we went through a week of you doing the dragon genetics. You sat there and you worked it out. You used popsicle sticks. You flipped them. You said, which one is it going to get? Is it going to get the big T or the little T? Is it going to get the big A or the little A? You did it. Then you looked at the percentages. Did you not? Or did you? Maybe you didn't. What's going on? Somebody explain to me. What's going on? What's going on? Hello? I'm talking to you. Did you do that last week or not? So we wasted Friday, according to you guys, which is cool. I'm sorry about that you feel I wasted Friday. What about the rest of the week? Where's your dragon genetic stuff? Did you upload it?
Yes.
And you did it? So now you answer me. If you did it, and you knew what you were doing, if you didn't know what you were doing, why didn't you ask a question then? You knew what you were doing. You're having the DNA. I'm asking you how many of each of these, first of all, at end of meiosis, how many haploids do I make? 4 is correct. What goes into each one of those 4?
1 T and 1 A, right?
1 T or 1 A, correct. Correct. 1 T and 1 A. Why? Because I have 2 T's and 2 A's, and now I'm trying to make half. Is that clear? So what I'm hearing is that we first start off with a confusion of how many genes you have. Now we've settled that, correct? Second thing is people didn't understand what foil is and why. Well, I think that's obvious. You're not taking what you experienced in the dragon genetics and applying it to making these sperm and these eggs. I don't understand where the misstep there is. You have to help me understand this. Where is the ambiguity? Where is the confusion? You went through and made gametes in dragon genetics. You made babies. You did it. You made babies. You chose wings or no wings, horns. I mean, if you did it yourself, if you participated in that, then you should have some understanding of what's going on here. So what's going on? Well, you got to make a baby, but before you make a baby, what do you have to make? What's the word?
Before you make the baby,
You have to make gametes. Everybody told me your dragon genetics were complete. You told me you understood, but now you're applying it to Punnett square and you're confused. Why? It's the same thing you've been doing all week. What is it you're trying to make? Gametes. What are gametes? Sex cells, which are what? Sperm or egg. 4 sex cells. Sperm or egg. Having 1 A, 1 T. Is that confusing to anyone? For each parent. For each parent. And you went through and you looked at and answered questions on this topic all week. So now, what do we have here? How do we do it? That's right, we would use FOIL, which is what? First is a big T, big A. Big T, big A. Next is what? Next is big T, little a. I feel like this is something you should know. I feel like this is something you did for an entire week, did you not? Am I wrong? If I'm wrong, you've been very vocal up to this point. So you have worked on this all week. So it wasn't an unreasonable expectation for me to expect that you would be able to do this, right? Is that correct? Okay. And then what is the next one? The next part of FOIL is little t, big A. And then what? Little t, little a. And because both parents have the same genotype, they can both make the same combinations. Is that correct? It sounds like everybody's frustrated because I keep asking, is that correct? But then people tell me they don't understand. And I'm wondering, is it you want me to do the work for you? Or is it you don't want to spend the energy thinking? Or is it you really don't understand the connection between the concepts? I'm finding it difficult to understand what your problem is. I can't help you if I don't understand what your problem is. Right? So you need to be clear, what is the problem here? What is the problem in the understanding? What is the problem with this content? So these are the two possible combinations. What is a Punnett square? A Punnett square is nothing but what different gametes are you combining? You're combining big T and big A, or big T and little a, or little t and big A, or little t, little a, or and big T, big A, and big T, little a, and little t, big A, and little t, little a. Where'd I get all that? Here. There it is. The gametes. One set is the sperm, the other set are the eggs. That's it. You did this for a week. You made them. You looked at all the different combinations. You put down the fractions. You answered them in words and in fractions. You did the math. Where's the misconnection here? Help me. What's going on? You sit there quiet now. You're quietly sitting there. Why? What's the problem? Where are you misunderstanding something?
I mean, I get the FOIL part. I get all that. I could do that. Not a problem, but like finding the percentage and the ratios is like throwing me off.
Why? You did it during the lab.
Well,
What's the difference between what you did 3 days ago, for 3 days last week, or 4 days last week?
it didn't ask me for the ratios.
But it just.
It did. You had to answer ratios last week as well.
Really?
Yeah.
Yeah.
But let's go ahead and do it. What part of, you're tired of what? Doing it? Is it just too repetitive?
What you mean? You repetitive like.
Are you tired because you know it and you're bored?
Yes.
I can understand that. If you've been paying attention, you've been doing your work, I can understand that, but we all have different mindsets on what preparation means and what it means to get ready. And also there's different levels of understanding. Some people may have done it and tried their hardest and still don't understand. I can understand that. But I get also a feeling that some people aren't paying attention. And I get that feeling very strongly. And some people pay attention but don't want to practice. I think that's a real big problem as well. Well, you may not need to practice because it sounds like you know it. I think some of your colleagues might need some more practice. We did this. This is what we did. I posted this. We did this in class. We counted up the ratios. And you did this in the lab for 3 days. You counted up ratios. They asked you fractions. They asked you ratios. So if you do a Punnett square, and they ask you for genetic ratios, they ask you, what is the distribution of the gametes? Well, as we did in class, we would say you have one of these. Is that correct?
What do you mean?
Big T, big T, big A, big A. There's only one. Do you agree?
Yeah.
And there's only one of these. So already that tells you, so if you listed the possible outcomes, right? Then you could count them up. You could say 1, that would lead to what? You got 2 of these, big T, big T, big T, big T, big A, little a. So that's 2 of those. What is going on? Big T, little t, big A, there's 2 of these. So there's 2 of those. Big T, little a, little a. How many of those do you have? 1. I mean, is this making sense?
Yeah.
If I have big T, big A, little a, I got 1, 2, 3, and 4. I mean, I'm just counting. These are ratios. Big T, little a, there's 2. Little t, big A, there's only 1. Is that correct?
Yes.
Little t, big A, little a, there's 2. And then there's 1, little t, little a, little a. Out of how many? How many do we have? What's a 4 by 4? How many different possibilities? 16. So this is 1 out of 16. This is 2 out of 16. This is 2 out of 16. This is 1 out of 16. This is 4 out of 16. Oops. I don't know what. 16. 2 out of 16. 1 out of 16. That's right. 2 out of 16. 1 out of 16.
So would you reduce them or just leave them like that?
You can reduce them. Always. It all depends on what your answer choices are. They rarely ask you this, though. They rarely ask you this. I think if you look at it, they'll really ask you about what? Phenotypic ratios. What they ask you about is phenotypic ratios. These are genotypic ratios, right? If you remember correctly, what a genotype is the types of genes the zygotes have, the babies have. You did this in the lab. So, I did this with you in class, and I said if you have 2 heterozygotes, what you end up doing is you end up getting a 9 to 3 to 3 to 1 ratio, right? Where you get 9 of them are going to be, I don't know what T is here, is tall, where T is tall, and what's the other one? Little t is short, and then big A is what? Terminal flowers, I think.
No, that's
No, A is auxiliary.
yeah, then the other one.
I'm sorry, auxiliary. And little a is terminal. That's my understanding. So you have 9 of them, 9 of these, 9 out of what? 9 out of what? 16 are going to be tall and have auxiliary flowers. 3 out of 16 will be short with auxiliary flowers. In other words, 1 of the recessive traits and 1 of the dominant traits. 3 out of 16 are going to be tall and terminal. Notice this one has the dominant auxiliary, but the recessive short. This has the dominant tall, but the recessive terminal. Phenotype, these are phenotypic ratios. And when we're dealing with what was I talking about? When we're dealing with the last one, which is 1 out of 16, that means you have recessive both short and terminal. Only 1 out of 16 will be short terminal. Those are your ratios. Those are your fractions. I don't think that's hard. If you have the Punnett square, and you can read this, T is tall and A is auxiliary, you can read it and count. It's just counting out of a total of 16. Or you can memorize 9 to 3 to 3 to 1, whichever one you want. Is that clear to everybody?
Yep.
I've gone through all this. I feel like I'm wasting time going back over it again, only because, to me, if we've gone over it, to go over the entire thing is a waste of time. But it seems like some of you needed this. I question why. All right. So again, does everybody understand all that? Now this is when you have two heterozygotes. You see how complicated and long it is. I've gone over all the different possibilities in class previously, but let's talk about what would happen if you had big T, big A, big T, big A, little a, and you crossed it with big T, little a, little a. Now what's the difference between the two?
One homozygous, one not homozygous, little.
Well, what are the different? This one you did this in the Dragon Genetics. So go ahead and open up your Dragon Genetics labs and take a look at what we're talking about when you're looking at. Let's see, where was it? Where you look at page 2, what the difference was in the gametes between the father and the mother. On the second page of the Dragon Genetics lab, and by the way, it was repeated again later, twice. I think you see on page 4, it was done again, mother and father, where the father was homozygous for everything and the mother was heterozygote. And it was done on page 2 again. So when you're dealing with, when one pair has limited genotypes, that limits the types of gametes that they can produce. So now, when you have limited genotypes, you limit the amount of the number of the different kinds of gametes you can produce. When we look at this one, how many different kinds of gametes it's going to produce 4, and you see that if you did page 2 of the Dragon Genetics. Are we all on Dragon Genetics? Do we all have it out? All right, we're looking at page 2. I see some of us are not. So page 2, if you did that, you saw what happens. Then on page 3, they have you walk it out and actually write down, look, father can only make how many different kinds of sperm on page 3? 1 kind of sperm, because he only has recessive genes. Mom can make how many different kinds of eggs? 4. Do you not see that that is a Punnett square? Do you see that or not? It is a Punnett square. And then you flip it over on page 4, and there it is again. And there again, they're asking what? How many babies with this genotype out of the total? Look at all the different times they ask you for fractions. Do you see that? How many different times they ask you for percentages and fractions? So when someone comes in after a week of doing something, we have a weekend off, and they come in and they say, did we do fractions? What does that say to you? They haven't really processed this. They haven't, whatever that means. Some people processed it on the spot. They spent the time last week, so they relaxed over the weekend. Some people didn't do that, spent the weekend and caught up. Others didn't do anything all week and the weekend. And they're coming in confused. They don't even remember what they did last week. Okay, so here we go. How many different kinds of sperm can this parent make? I think everybody should know already. You tell me. If you took notes, you should be able to tell me right now. What are they?
Big T, big A. The same with the last Big T, little A. Little T, big A. Little T, little A.
Perfect. Exactly what we said before. You can see it. It's not hard. But if you need to do FOIL, if FOIL helps you, big T, big A, big T, big A, big T, little t, big A, and little t, little a. Now, how many different kinds? We're going to go ahead and we're going to do what you did on page 2. We're going to do it here. It's going to make 4. We're not including crossing over, because crossing over would increase the variation, correct? So, if you did FOIL, what would happen? Big T what? And then big T. So big T, little a, big T, little a. And what's this one over here? Little a, little t, little a. So there's only 2 different kinds of eggs this parent can make, or a sperm, or whatever it is. So really, you could do a Punnett square. You could do a 4x4 Punnett square if you really wanted to. You could put big T, big A, big T, big A, little t, little a, little t, little a. Why don't you do that? Why do you get rid of half of these?
Because it decreased the work.
There's only 2 different kinds, and they're going to be the same proportions, right? It'll be the same. There'll be different numbers of them when you do the math, but they'll be the same fraction, the same percentage. Does that make sense? All right. So you can do that. You can just get rid of it. Mathematically, it's okay not to worry about these duplicates. We don't have to do it again for the duplicates, which is the whole point of the lab when they had you walk through those activities. Big T, big A, and little t, little a. But there are 4 different kinds of gametes the other parent can make. So we should make sure that we add those. And when we combine them, we produce big T, big A, little a, big T, little a, big T, little t, big A, little a, big T, little a, little a, big T, little t, big A, little a, big T, little a, little a, little t, big A, little a, little t, little a, little a. So now you don't have 16. How many can you make? 8 total. So your fractions they're different bottom number, but the top number is still going to be whatever you count. Things are simpler. Are we clear on this? This should have been clear at the end of Dragon Genetics. If you answered all the questions the way some of you've said, you've done, and you didn't ask me any questions, then there should be no issues. So let's take a look at these fractions. Big T, when we look at big T, big A, little a, that's one kind here. There's only one of those. Again, doing the fractions, 1. I feel like, honestly, I just keep doing this. And here's my problem. I see some of you staring into space. I see some of you not writing. I've been through this already once, the whole thing. I've explained it before we even did Dragon Genetics. We went through Dragon Genetics. I got a lot of kids that understand this. The kids that don't get it are the ones that aren't paying attention now. And I feel like this is not productive. This is what I'm feeling. I'm looking, and it's just not being productive. I feel like I'm wasting our class time on this. I would tell you to go to tutoring, but some of you don't go to tutoring. Well, most of you don't go to tutoring. Only 5% of you go to tutoring. And so I would say, come and see me after class if you're confused, but you don't do that. So now what? What are we going to do? You should have done this in the lab. Obviously, some of you didn't get it when you did in the lab. You didn't get it when we covered it in class. So now what? So you have to do, I gave you a chapter reading, didn't I? You all have a chapter, a book chapter, you can read it. You can do that too. Any one of these things will work, but you got to do something. All right, so I'm not going to go through all these different options, but you can figure that out. Phenotypically, though, I will go over the phenotypes really quickly, and then I guess I can make some use of today's time. Obviously, we're not getting any of these growth conferences done today. We'll try again tomorrow, when I'm going to expect that you're going to be able to do these problems tomorrow. Is that a reasonable expectation that this class is going to be able to do this tomorrow? Is it a reasonable? I need you to pay. I hear some of you saying yes. I see you nodding your head. I see some of you that literally said you did not cover it last week are not even paying attention to what I'm saying right now. So I'm asking the entire class, as everyone in this class right now, is it reasonable for me to expect that either you can do this tomorrow while I do growth conferences with each individual student, or you're going to get help from me or from a tutor from a book, and you're going to be able to figure it out tomorrow in class? Is that reasonable?
Yes.
Okay. So, that being said, let's go ahead and do the phenotypic ratio. I think class is over, what, in a minute?
Yeah.
Okay, well, there goes all that wasted time. I want to tell you before we go on, what if I had a parent?
Okay.
Pay attention, because this is a homework question for you to figure out. What if I had a parent that I knew, this is a parent, I didn't know the genotype, but I knew the phenotype. I knew it was tall, and that it was, and axillary. Could you hold on a second before you put anything away? I know it's literally just now turning time to class. Sit down. You putting stuff away before class is over sucks up class time. It disturbs the class flow. And I don't know, I've said this how many times? More than once, right? 10, 20 times? I've asked you not to put things away until class is over. I could have done this, I could have gave you this assignment, this food for thought in the last 30 seconds, but I had to stop to discuss you putting stuff away before class is over. That's very disturbing to me. Is that not reasonable? All right. Obviously, we're not going to be able to do that. Have a good day. So now, I want to remind you that when we have this simpler version of Punnett squared, it's simpler because one of the genes here, one of the genes and one of the parents is homozygous recessive. If this parent were short with terminal flowers, in other words, little t, little a, if this parent was homozygous for recessive for both of these, then there would only be one row here. Does that make sense? Because there's only one possible gamete it can make. Yes, it'll make all four. Meiosis still makes 4 cells, but all the genotypes will be the same. Now let me ask you something. What if you didn't know the genotype? What if the parent was tall and axillary? So what does that mean? What does that mean, the parent's tall and axillary? Has at least one dominant for both, right? Has big T blank, big A blank. Do you know what that second letter is?
No.
Why not?
That's the same homozygous.
Yeah, but I think it's important you listen. What's this now?
That's the same homozygous.
Right, you don't know if it's homozygous or heterozygous. Well, how can you tell? If you see something that's tall and axillary, is there anything you could do? I'll give you a clue, the answer is yes. Is there anything you can do to figure out what that second letter is?
Big T.
How do you know it's big T? How do you know it's not little t?
Because it's all.
Look at the combination.
Because of the, yeah, because of the combination of the square chance.
Is it all dominant? Would you have it
Which means if this tall.
Have it breed with what?
with, or with one that's all
Homozygous dominant?
recessive?
Homozygous recessive. So if I do this, if I cross it with somebody like this, then I can tell. And here's how I can tell. This is, by the way, this is called a test cross. A test cross is used when you see something that has the dominant traits, but you're not sure what the genotype is. We call it a test cross. So if this particular parent, that makes life easy. There's only one gamete it can make, right? Little t, little a. Does everybody agree with that? Well, if this parent, there's two possibilities, right? That's either the parent is big T, or the parent is big T, little t, correct? Either parent's big A, or it's big A, little a. Only two possibilities. We know it's big A, and we know it's big T. So it's one or the other. There's an or involved. So either you have little t, little a, crossed with big T, big A. Is that correct? Because if this is homozygous big T, big A, there's only one possible gamete it can make is big T, big A. Correct? If the parent is big T, big A, little a, then the parent can make four possible different gametes. It can make big T, big A, big T, little t, big A, and little t, little a. Correct? It still would only be crossed with little t, little a. So if the parent is homozygous dominant, then what's the only kind of plant, 100% of its progeny is going to be what? Dominant. It's 100% of the zygotes, of the progeny, the children of this organism is going to be a dominant phenotype. It's going to be tall with axillary flowers. There is no other choice because it had the dominant traits, the dominant genes. Is that clear? It's the only gamete this thing can make. If, on the other hand, if there's even one that's short or with terminal flowers, then you know something, don't you? Big T, big A, little a. Big T, little a, little a. Little t, big A, little a. And little t, little a, little a. If you get even one of these that's short, just one baby plant that's short with terminal flowers, then you know what? You know mom, or you know this parent has what? Has a recessive of each of the genes. You know that. See, that's the trick. That's the trick of a test cross. If you get 100% of the progeny are dominant phenotype, then you know there is no way that this parent, right, could have a recessive gene. Now, of course, like we know, this is probability, so you're probably going to want to take 1,000 different examples, right? You can't do one, because you could always get one that could end up being tall, could have the dominant phenotype, because there's a 1/4 chance you have the dominant phenotype, and then you have, 75% chance of getting a recessive of one of the two traits. Is that clear? So that's what we call a test cross, and it's important when you're doing, it's important for various reasons, but when you see a phenotype, you don't know what the genotype is. You have to do a test cross to figure out what, or you have to sequence the DNA. One of the two. The test cross, again, if you just have one recessive outcome, then you know that that parent has the recessive genes. If you get none, then you're not 100% sure, because it could just be luck, right? Like flipping the coin 50 times and getting heads 50 times in a row. It could be that it's just pure luck that you just keep getting this outcome instead of any of these. But the key is that if you just get one that has a recessive gene, recessive phenotype, then you know they have that recessive gene, because it's the only way to get that recessive phenotype. Is that clear? All right, so what's your job? The rest of the week, you have to do two things. There's going to be two objectives. Listen, write this down. Number one, read your chapter and take notes. It's important because we've been talking about dominant recessive, but now there's other kinds of inheritance. And the second thing that you have to make sure you do is complete the practice problems. On your outline of, here in this outline, I believe you needed one of these, right?
I need one.
There's more up here if you need one. These, in this outline, on the back, you'll see different kinds of inheritance. If you haven't read the chapter, you won't understand what's going on. Tomorrow, Wednesday and Thursday, I want to do growth conferences with everyone in this. I hope to finish early, but if we have to keep going, then we got till Thursday in my plans to get these growth conferences done. I want no one surprised about their grades. I want to make sure every grade issue is resolved. So that's what we're doing the rest of the week. Good luck.